I'm taking up 90% of it on one of the panels, but I think that'll be okay.
Not for everyone. We're talking to the little camera.
It's an
Did it
owl.
move? Yes.
So,
I think when it's just me talking, I have been doing
We got
the long
yeah.
panels, but maybe when I go to present it,
That
it
was when
changes.
Okay. I think it'll be good.
You got your grandmother?
Yes.
Looks like that.
Yes.
Okay. So,
Okay. I'll do the first.
and I'll start to introduce. Okay, do we have a, is this working for, can you check if this is working for the online people?
Oh, yeah, that's
Did
true.
you check sound?
I checked it. Can someone who's on the Zoom type in the chat if you can hear us? And Megan, maybe let us know.
We can hear you well.
Thank you. Okay, then let me welcome all of you in line and online in the room, the ones of you who fittingly weathered the storm here to come to Eli's defense party. No, the defense, the party is later. His public portion of his oral defense for his PhD thesis. And thank you for all of you who stayed at home, but participate online through Zoom or the live stream for staying safe. So Eli is, this is his first oral public portion of his defense of his thesis. He came to NC State in 2016, a few years ago, after he graduated from University of Chapel Hill and Duke University, yes, both as an undergraduate. He had a Robertson scholarship that allowed him to attend both and funded it. And he didn't do the same thing at both universities. He studied biology at Duke University, and there's our last committee member. And he studied linguistics at University of North Carolina, Chapel Hill. These two different aspects continue. So Eli speaks several languages, including fluent, Spanish and Swahili, and conversant Mongolian and Mandarin. We will not test it today, but I'm sure there are some people online. After he graduated from Duke and UNC Chapel Hill, he tried to get away from the triangle and went with a, what was it? The Fulbright Fellowship to Mongolia for research here, looking at how seeds from some plants in Mongolian southern Gobi Desert germinate, and then came back, made it back here. He did a few other things trying to get away from the triangle. None of them worked permanently. So he's still here and started his graduate studies with us. He got first a provost doctoral fellowship, which is a really nice acknowledgement of his previous achievements. And then we tied him down in the Ag Biofuels with an Ag Biofuels fellowship. This is an NSF research fellowship that is interdisciplinary. Fred, one of the committee members here, is the PI on it, which tries to really look at research in a more in agricultural research in a more interdisciplinary aspects, especially at the issues of genetically engineering of crops. One of the requirements for the Ag Biofuels Fellowship, besides having faculty member from Ag Biofuels on it, is that he has to have a thesis chapter in a discipline that is not related to his major thesis. So he has a major section on the economics of lupin in North Carolina and the US worldwide. Mostly US. And so he will probably not get to present it. So I ask you to present it to the committee afterwards. But if you have questions, you can still ask about it. Lupin is very pretty and useful. So during his time, Eli was was a challenging project, as you will see in a minute, that Eli took on. But he managed to really get it to a very exciting point, not without many hurdles. So the hurricane in the background is pretty fitting. During his time, he was able to publish 2 papers, one together with the Ag Biofuels cohort on lessons for a secure future, evaluating diversity and crop biotechnology across regulatory regimes. And a second one that was just accepted this week, a high throughput detection of tDNA insertion site for multiple transgenes in complex genomes. Long title, complex research, but very important for industry to identify where transgenes are in complex genomes like polyploids. He also was able to secure a patent as a co-inventor on this topic of reestablishing avuscular mycorrhizal symbiosis in non-host plants. Yeah, that's kind of so far. He will publish a lot more papers from his thesis starting Monday. So I want to thank the committee. As the representative for the graduate school, Dr. Henry Schaefer, thank you. Henry doesn't have to do these things anymore, but he's always happy to hear about synthetic biology. Dr. Ross Susani, her flight got canceled, so she shipped up her kit and made it. Dr. Fred Gould from the Ag Biofuels, but also in the area of agricultural engineering. And we have Mark Kibeda, Dr. Mark Kibeda here, who is the expert really in fungal biology and symbiosis. With that, we're going to start. Eli will give a 45-minute presentation. We will take then questions from the public. Everybody online is monitored by ASA, so if there are any questions, we will try to get Eli to respond to them. We will only have 10 to 15 minutes. After that, the public session is over. We'll have 5 minutes break, and then the committee meets in a closed session. Anything else? There are a lot of other skills Eli has here. He's a great baker. So every lab is happy to have. So with that, all yours.
Right. So thank you, Heike. Thank you, everyone, for coming out on this beautiful day. And if you're watching online, again, please try to type any questions in the chat. We have people watching, they'll repeat it, but that'll let us answer as many as possible. So the title of both my thesis and this talk is Genetic Engineering to Retrace the Evolutionary Loss of Arbuscular Mycorrhizae with Implications for Sustainable Agriculture. And that is quite a mouthful. So before we go any further, I'm just going to briefly tell you what mycorrhizae even are. They are a symbiotic relationship between plant roots and fungi in the soil, which help the plants take up nutrients and also fortify them against a number of stresses that can occur in different contexts. These are an often overlooked but fundamental aspect of plant biology. Most plants have mycorrhizae. Having said that, a minority, about 5% of plants, have lost their mycorrhizae and not replaced them with any other type of intracellular symbiosis. And although they're a phylogenetic minority, they turn out to be fairly important. Yes, Megan?
The audience is wondering if you're able to share your screen on Zoom so that they can see it on their laptop.
The people on Zoom cannot see. All right.
Please record.
It is recording. Can the people on Zoom see now? Great. Thank you, Megan. So although these are a phylogenetic minority,
Yeah,
don't worry.
Megan, can you-
We have people on the Zoom. Although they are a phylogenetic minority, they turn out to be important in agriculture because we've domesticated quite a few non-mycorrhizal plants. So notably, the whole mustard and amaranth families are not mycorrhizal, and that includes major commodities like canola and sugar beet. That's the canola growing region of North America. It's quite big. But If that's too abstract for you, if you're here in person and you had the snacks, you just ate about 10 non-mycorrhizal plants. So you see these in the grocery store: broccoli, cabbage, kale, quinoa, beets. All of these are non-mycorrhizal. And if you work on Arabidopsis, in a lot of regards, you're not really working on a generic model plant. You are a non-mycorrhizal plant specialist, because Arabidopsis is one of those 5%. So it has a big impact, and understanding both mycorrhizae and their loss could be quite important. So the way the talk will go is I'll give a more detailed overview of exactly what mycorrhizae are and how they work. Then I'm going to focus on my most recent piece of research, which is a transcriptome of Arabidopsis expressing IPD3, which is a gene that's essential for mycorrhizae. That turns out to have implications for defense. And then, as Heike said, in the last few minutes, I'm just going to give a very quick overview of this lupin work, and I'll go into more detail with the committee, but happy to answer questions on this or anything else that you may happen to have already known about my research. So from now on, when I say mycorrhizae, I mean arbuscular mycorrhizae. There are multiple types, but arbuscular mycorrhizae are, in a fairly literal genetic sense, the granddaddy of them all. And the way they work is that fungi grow from the soil into the root, into deep cell layers, then through the walls of living plant cells, where they form the essential structure of the symbiosis, which I've highlighted in green, which is the arbuscule, which is a very dense reticulated structure with high surface area where nutrient exchange can take place. When I say that mycorrhizae are a fundamental aspect of plant biology, and most plants have them, it's because they're inherited from the single common ancestor of all land plants. One way to demonstrate that is by looking at this, which is a liverwort, a primitive land plant. It has no root, shoot, stems, or leaves. It's just sort of a flat sheet that lays on the ground. And what plays the role of a root for this flat sheet is its bottom half, just the part closest to the ground. Highlighted in red is a liverwort that's been sliced in half. Note that the cells are empty. Highlighted in green is what you get when you grow some liverworts with mycorrhizal fungus. They get colonized with arbuscules in the portion of this sheet that works as a root for them. So the way you can think about it briefly is plants have had mycorrhizae since before they even had roots. And everything that's happened in their evolutionary history since then started from an initial mycorrhizal state. Something else important to understand about what we now know for mycorrhizae is that while we primarily see them as a nutrient-related trait, we're increasingly understanding that they have really important impacts on biotic and abiotic stress. So the presence of mycorrhizae improves drought resistance indirectly and by funneling water right through the fungal hyphae to the plant root. They protect the plant against both heat and cold stress, toxins in the soil, and through a phenomenon known as mycorrhizae-induced resistance, or MIR, against a whole range of pathogens and insect pests. And not to put too fine a point on it, but this is all climate change stuff, right? We're really worried about this stuff, and we don't have good answers for it. So I think, at least in the context of agriculture, we could consider changing the way we think about mycorrhizae to a trait that is possibly of primary interest as a stress resistance trait, as opposed to one that is of primary interest as a nutrient uptake trait, although both are, of course, important. One reason it's always been seen as a primarily nutrient-related trait is that it is very closely tied to the amount of nutrients in the soil. So if you add fertilizer to soil where a mycorrhizal plant is growing, you will strongly reduce the amount of mycorrhization. And this is a bit of a paradox for agriculture, because fertilizer is pretty much our most accessible agricultural innovation. If you're not using fertilizer, it's pretty much just because you can't get it. But when we use fertilizer, we inhibit the formation of mycorrhizae, which prevents it from protecting against any of these stresses that may occur later on in the life of the plant, and which we don't have very good solutions for, not without building a whole lot of infrastructure or doing a whole lot of sprays. So this, to me, as a genetic engineer, is a reason to consider potentially tinkering with the way these work at the genetic level to try to make them fit in better to sustainable production. Right. Another thing we have recently learned about mycorrhizae is that they are directly controlled by another ancient plant feature, which is the phosphate starvation response. This is as old or older than mycorrhizal symbiosis itself. Its core function is through a protein called PHR, which is a transcription factor, which in the presence of phosphate is bound up by a different protein called SPX and prevented from regulating genes. In the absence of phosphate, it turns on all the genes involved in the mainline phosphate response. In just the past couple of years, it was discovered that that includes at multiple points the genes that control mycorrhizal symbiosis. So it makes perfect sense that in the current state, we see suppression of mycorrhizae by the addition of fertilizer because they're subordinated to a fertilizer sensing system. So unless we change some things, we are going to be stuck in this fertilizer regulated state. When it comes to non-mycorrhizal plants, we understand at the genetic level what's involved in their loss. So what you're seeing here is for the list of plants on the left, a blue square is the gene that's conserved, a red square is a gene that's lost. And just focus on the intersection of the green box and the red box, which is Arabidopsis thaliana. There is a small handful, say 9 to 12 genes, depending on who you ask, of genes that are always lost when you lose mycorrhizae. And there's 3 subsets of those genes. Some of them relate to nutrient exchange and actually making arbuscules, unsurprisingly. Some of them belong to something called the common symbiosis pathway, which is a bunch of sensor proteins and transcription factors that act to turn on the functional genes, basically like a remote control. And then the third class relates to lipid biosynthesis. And this really came together since I started graduate school. It was in 2016. So when I started out, people kind of thought mycorrhizal fungi ate sucrose. Then we had all this phylogenetic work saying that apparently lipid synthesis is really important for the ability to have mycorrhizae. Simultaneously, some functional genetic work showed that the primary carbon source for mycorrhizal fungi from the plant is a very particular type of lipid called monoacylglycerols. You don't need to worry too much about the details of this slide, but the things that I've circled are generic fatty acid synthesis enzymes leading up to monoacylglycerol synthesis and transport proteins down towards the bottom of the slide. And those, that last class of genes that are lost when you lose mycorrhizae are specialized copies that do the same biochemical function as these genes, but in different cells and in response to the presence of mycorrhizal fungus to provide these lipids to them for their nutrition. So bear those facts in mind. The questions that really drive my research are why, if mycorrhizae is as beneficial as I've made it sound, would plants lose them, right? The fact that these were lost at some point in the ancestry of quite a few plants indicates they must have had some cost or their absence must have been beneficial. There's a few theories in the literature. A prominent one, which has a parallel in nodulating symbiosis, is that maybe they just cost too much carbon. So making all of those lipids and sending them to the fungus may have been too much of a burden on the plant photosynthesis. That should prick up your ears because right now we have a lot of extra carbon around. We also have a lot of extra environmental stress around. So a trait that might let you swap carbon for improved stress resistance would be a very high interest. Another possibility is that there was some sort of trade-off between symbiosis and defense against fungal or bacterial pathogens. So there's the general phenomenon of MIR, where mycorrhizae seem to protect plants, but at the specific level, it's also true that some symbiosis-related genes in some plants in interaction with some pathogens cause greater susceptibility. And it's possible that at some point in the particular ancestry of non-mycorrhizal groups, that was just enough to tip them into a point where it made more sense to do away with those genes altogether. The last and least controversial, but also least extensible reason plants may lose mycorrhizae is a big change in lifestyle. So for example, plants that become aquatic lose their mycorrhizae. But I don't think we have the evidence to try to put that on a gradient and say, well, plants that move from the mountains to the coast are going to have a certain change in their mycorrhizae. But if in the context of agriculture, the way I think about it is for a loss that occurred quite a long time ago. In the Brassicaceae, for example, they split off from their nearest mycorrhizal ancestor 60 to 70 million years ago. They went around evolving and radiating since then. They're now in very different habitats. They have different forms. And for the ones we've domesticated, they're in incredibly different habitats, living very different lifestyles for a very different purpose. So in their new contexts, is it still advantageous for these plants to lack mycorrhizae, or are they just compensating for something that occurred in their ancestry and that they can't get back because of all those lost genes? If it would be advantageous for them to have mycorrhizae, as a genetic engineer, the question is, how would you go about putting those back? And the way I tried to answer this compound question is by inserting a particular gene called IPD3 or Cyclops, highlighted here. This is one of the genes in the CSP, one of those remote control genes. And the way IPD3 works is that upstream of it, there's a whole bunch of signaling that takes place in response to the presence of the fungus, leading to activation of this protein called DMI3, which phosphorylates, and I'll explain that in a minute, IPD3. IPD3, when phosphorylated, acts as a transcription factor and regulates many downstream genes for these functions that enable symbiosis. So here, I'm showing it kind of linearly regulating some genes related to lipid synthesis, but this is actually a network down here. This goes back and forth, and there's a lot of crosstalk and additional targets. One thing that will tell you how powerful IPD3 is and why it was a good transgene candidate is that if you use a version of the protein which is engineered to be permanently switched on, you get a transcriptional and phenotypic response in symbiosis models that looks as if a symbiotic partner was present, even when it's not. And so if we're going to put a gene into a plant that we know has lost a lot of other symbiosis genes, this seems promising because it suggests that IPD3 can substitute for the absence of a whole bunch of additional genes that might be missing before it. So we could sort of get many for the price of one. Another reason to be interested in IPD3 is we know it's a specific intersection point for some of these other conserved plant functions. So IPD3 is one of those genes recently found to be regulated by PHR. PHR is, of course, conserved and very well understood in Arabidopsis. IPD3 also co-regulates genes with PHR, so we may see an interaction if we put it in parallel. Also, lipid synthesis is now known to interact with some of the near targets of IPD3 via wrinkled transcription factors, which are highly conserved in plants, including Arabidopsis. So if we put in IPD3 right near this intersection point with a lot of native Arabidopsis biology, we may discover some connections that it still retains in Arabidopsis. And of course, the last thing that may happen is maybe we just forget about everything we know about symbiosis. And by putting IPD3 into Arabidopsis, we might discover, for example, what it was that made it disadvantageous in the past. We might discover unknown intersections with other parts of plant biology. So the way the synthetic activation of IPD3 works is this. It has two domains, shown in red, the DNA binding domain, in blue, what we'll call an activation domain. In its natural form, labeled MT-IPD3, MT stands for Medicago truncatula, which is a symbiosis model. IPD3 is phosphorylated by DMI3. That means DMI3 puts a phosphate ion on a particular site on that protein. The phosphate ion is negatively charged, which causes the protein to physically change shape and exposes the DNA binding domain. That allows IPD3 to then regulate genes. The first way to synthetically turn on IPD3 is by phosphomimicking, which is a pretty well understood practice in the literature. It's also something that other researchers have already shown to work for this gene. All you do is replace the amino acid at the site that normally gets a phosphate put on it with one that carries its own negative charge. So that means the protein is permanently unfolded, the DNA binding domain is permanently exposed, and it always regulates DNA as if it had been activated. The last way to synthetically activate IPD3 is to just chop off the whole activation domain. And again, some great work by other researchers has proven that you can do this. So if you take just the final 45% of the protein, just the DNA binding domain, express that in a symbiosis model, you can see what looks like a permanently activated IPD3 version being expressed. So what I did is put all three of these versions of IPD3 into Arabidopsis under a constitutive ubiquitin promoter. And not shown here, as Heike mentioned, is the incredible amount of work it took to get this transgene construct right. So not just multiple versions of the construct, but actually across multiple species. But for the one that worked, I brought it to homozygosity across 3 generations of Arabidopsis. And when we got to that endpoint, what we saw was this. And this is a Western blot. It shows expression of the protein product of these various IPD3 versions. And what we see is that IPD3 min is the only one that shows up on the blot. It's highlighted with those little yellow carrots. If you're used to looking at blots, this isn't the very clearest one. That's because just like the genetic engineering, this is hard to get on a blot. It's a little blurry. There's some spurious bands. These are slightly different sizes. So we confirmed that what we're seeing here with the help of our collaborators in the Manuel Kleiner lab. So we did 3 things. First, we cut out these bands on the IPD3 min protein gel and sequenced them with mass spectrometry. That confirms unequivocally that what we're seeing is IPD3 min. Then we cut out the places on these other protein gels where the full-length versions should be, which would tell us if, for example, the protein was present on the gel but not being stained by the antibody. And the mass spectrometry there confirms that no, the protein is not on the gel. It is just not present. Finally, we did untargeted shotgun mass spec proteomics, where you just take a full protein sample and look at everything you can find. And in that case, shown on the bar graph here, in the root and the shoot of IPD3 min plants, we find the transgene product pretty abundantly. It's actually in around the top 10% most abundant proteins. For every other version, it's a big fat 0, absolutely nothing. So this cannot rule out that we are still failing to extract the full-length versions, but barring that, the suggestion, since we find RNA expression of these constructs, is that the RNA is being very aggressively silenced by something in Arabidopsis. And since it's specific to the full-length versions, that would suggest that the silencing targets that portion of the transcript that's removed in IPD3 min. So that's a very interesting prospect. MicroRNA regulation is not unknown in mycorrhizal biology, but not specifically targeting IPD3. That's an interesting lead to follow up. The practical outcome for now is that we're going to focus on IPD3 min because that's the one where we know for sure we have protein. We looked at these plants for various aspects of their phenotype, just to see if their growth is changed in any particular way. And the most striking thing we found, if you look here, compared to the wild type Paulo on this side, 3 out of 4 independent lines that you're looking at are shorter. And in Arabidopsis, all the height comes from the flowering stem. So the height that you're seeing here translates to a difference in flowering biology. And you might notice they have fewer seed pods and the seed pods are less developed. Line 303, I might want to save this for the Q&A, but you'll just note it's not the same as the others. So when we looked at the timing of flowering onset with data, we confirmed what we can see with our eyes. So for 4 out of 5 independent lines that we looked at, there was a 4 to 5 day delay in the onset of flowering relative to wild type. And for Arabidopsis, 4 to 5 days is an eternity. This is a big difference. You'll note line 303, it actually is a little bit delayed relative to the wild type, but not to its null segment. Just keep it in the back of your mind. So a strong effect on flowering. The other thing we noticed is that the roots of our transgenic plants are just violently bright bubblegum pink. This might be our selection marker, which is a red fluorescent protein. Sometimes you can see those by eye. In our experience, you can't see them this way, but it's possible. What really came to mind was anthocyanins, because these plants might be stressed, which would cause them to accumulate anthocyanins. And with the help of our collaborators in the DeUssier lab, we spent some time really trying to extract and quantify anthocyanins. We also looked for carotenoids, and what we confirmed is that this isn't anthocyanins. We get nothing. So we don't know what's causing this color. We're not going to focus on it. I would love to find out, because one thing I noticed is that the color correlates with the strength of the above ground phenotype. So it's strongest in the short, low flowering plants. It's basically gone in line 303 here, which doesn't show the flowering phenotype. So if we just pause, it's fairly remarkable that 70 million years after this gene was lost, a gene which has no similar copies in Arabidopsis, which is considered to act only in a trait which has been lost from Arabidopsis, and whose known interactors have been lost from Arabidopsis, does something when we put it in. It affects a pretty fundamental part of plant biology, at least one part, in a strong way, which means it's acting as a transcription factor, it's interacting with a protein, but it's not null. And it's promising for this approach of trying to partially rebuild this lost pathway. So that was enough for us to look at its genetic effect using a transcriptome, which is a way of measuring the expression of all the genes in the genome. And the four main questions I want to explore for this are: one, what might IPD3 be directly regulating? A transcriptome can't fully tell you this, but it can tell you where to look. Number two, what is the effect of mycorrhizal fungus exposure on general Arabidopsis in the absence of IPD3? Number three, what is the effect of IPD3 expression in Arabidopsis in the absence of mycorrhizal fungus? And number four, is there any interaction between the expression of IPD3 min, by the way, IPD3 min, and the response to mycorrhizal fungus? So to answer the first question, we set up the transcriptome this way. And if you focus on the half MS side, half MS means fairly high nutrients, you can see that we have two independent transgenic lines. That's line 308 and 310. So one with a very extreme phenotype, one with an intermediate one, compared to wild type with and without mycorrhizal fungus. We also have a single transgenic line, line 308, compared to wild type with AM fungus on low nutrient media. Why wasn't the experiment factorial for low nutrient media? The answer is it was. The samples on low nutrient media didn't yield as much or as good RNA, and when we sent them for sequencing, a lot of them couldn't be used. So we're actually going to focus on the half MS side here. And to ask about the direct targets, I compared 5 different contrasts for differential expression. These are contrasts where IPD3 is the only difference between the control and the experimental. So for 2 independent lines, line 308 and 310, with and without AM fungus on half MS, and then for a single transgenic line with AM fungus compared to wild type. So this is across a pretty large amount of variation in other factors, but with IPD3 always making the difference. And when we do that, we get this big blobby Venn diagram, which you don't need to worry about other than this thing in the middle. 8 genes are always regulated in the same direction by IPD3 in all of these different contexts, which to me suggests they're good candidates to be direct targets of IPD3 for regulation. What are those 8 genes? I'm going to talk about 3 of them in detail. The top one is called TAR1. It's upregulated about 40-fold Conveniently, it was first characterized by the Alonzo Stepanova lab here at NC State. TAR1 and its homologs act in auxin biosynthesis. So it executes the first of two steps to turn tryptophan into indole acetic acid, a very important plant hormone. In order to actually get indole acetic acid, you need the second enzyme in that pathway, which would be a yucca gene, which is probably familiar to a lot of plant biologists. You should know that when I look into our transcriptome, for at least line 308, I see there is a yucca gene, yucca 9, which is also upregulated along with TAR1, although to a lesser degree. So it's a reasonable hypothesis that these plants are making more indole acetic acid somewhere. We don't know exactly where in the root it may be. The effect of overexpressing one or both of these genes is a shortened root with weird branching. And when we look at our plants on plates just informally, we see something that could be similar to this. We're going to have to look in quantitative detail to be sure, but definitely enough to be worth looking into. The next gene I want to look at is the unfortunately named cytochrome P450 family 76C2 enzyme. This is a very big gene family. This particular copy of it is unique to the Brassicaceae, which is odd that a gene whose loss, IPD3, is unique to the Brassicaceae, is regulating a gene whose presence is unique to the Brassicaceae. That's going to take some deep thinking to untangle. But at the more sort of thematic level, what this enzyme actually does is act in monoterpenol biosynthesis. Monoterpenols are volatile compounds which are involved in plant defense. They happen to be part of mycorrhizal-induced resistance already. So in general, we can squint at it and say, boy, it looks like this defense-related function is sort of parallel to what you see with mycorrhizae. But at the genetic level, this is a bit of a puzzle. Again, interesting enough to follow up. And the last gene I want to talk about is called FKF1. It's the only gene in this set that is downregulated, you'll notice. FKF1 is a circadian clock gene. It's shown in blue here. It is the protein that lets plants turn long length of individual days into rapid flowering over the growing season. It does that through protein-protein and transcriptional interactions with some really big names in plant genetics, with Gigantea, constans, flowering locust T. The outcome of which is when you grow plants under long day, they flower quite a bit sooner. Now, when you knock out FKF1, you get plants which are sort of dwarfed looking and flower later under long day length. My plants were grown under long day length. They're dwarfed and they flower later. So this is maybe a gene that's of interest for that function. We have to be a little careful with this because remember, we're looking at root genes. And this is a shoot function. So it's probably, IPD3 is probably also expressed in the shoot under the promoter we used. We need to look at it to be sure. But even if it is, this might kind of be a side effect of us overexpressing the gene in the shoot where IPD3 is not normally found. More interestingly, we should consider why IPD3 would interact with the circadian clock gene at all and whether it might have a root-specific function. And there's some implication of genes like FKF being involved in things other than just sensing day length. Sometimes they're involved in stress response, cellulose synthesis, so things to look at there. But we have to move on to the second question, which is how does mycorrhizal fungus exposure affect expression of genes in Arabidopsis? So this contrast is we add mycorrhizal fungus to just a plain wild type Arabidopsis. And the outcome is about 500 genes are differentially expressed. Of those 500 genes, when we analyze them using what's called gene ontology, which is a tool to look for patterns in the functions that are known for a big set of genes, we see things related to fungal and bacterial pathogen defense, osmotic and oxidative stress, responses to lipid, and suberin biosynthesis, among other things. So keep the defense stuff in mind. It's going to come back. But I want to take a second to look specifically at suberin, because suberin is a plant structural wax, which is made out of monoacylglycerols, which you might remember from earlier is what mycorrhizal fungi eat. And when we look in the transcriptome, what's circled here are the enzymes throughout that pathway that are differentially regulated by AM fungi in Arabidopsis. So the ones with the up arrow are upregulated, and you see genes related to general fatty acid transport and synthesis, but also at least one committed step for monoacylglycerol synthesis, which is GPAT5. There's some fine points here about which GPAT it is. Again, happy to talk about that later. And we also see transport of what would be monoacylglycerols across the cell membrane and the cell wall when we look deeper in the transcriptome. So very interesting that this same metabolic pathway, which would be involved in feeding the fungus, is expressed in a non-mycorrhizal plant, a plant where the fungus has not yet made it into the root, by the way, at the length of time we've exposed them for and in the context they're in. So we're going to look at the next question, number 3. What is the effect of IPD3 alone on Arabidopsis when no symbiotic microbe is present? This is the contrast. We express IPD3 in the wild type background. I'm showing you, by the way, results for just line 308, which has higher expression. The results for line 310 parallel this, but with a much smaller gene set. We see about 700 genes that are differentially expressed. And when we do gene ontology for those genes, we get something that looks very similar to AM fungus exposure alone. So we see bacterial and fungal defense, osmotic and oxidative stress, response to lipid, interestingly not response to suberin or suberin biosynthesis, and some things which are not specifically found in the AM fungus response, but which are related, like systemic acquired resistance and some of the hormone signaling related to it, like jasmonic and salicylic acid. We also see sucrose transport kind of moving around. It's hard to parse this, but we always pay attention when sucrose starts moving around. So if you think about what IPD3 does in natural host plants, when you express a synthetically activated version of it, you get a transcriptional response in the absence of microbes that looks like the plant sees a microbe, even when it's not there. This looks like we might be seeing something similar. The response to just expression of the gene with no fungus present has strong similarities to expression of the expression of native genes when you add fungus to just the wild type. So we can compare these two things directly. And when you look at these two gene sets side by side, of the 500 genes that are differentially regulated by mycorrhizal fungus exposure in the wild type, 83 are also found in the effect of IPD3 in the absence of fungus. Of those 83, 77 are regulated in the same direction in which they are regulated by exposure to AM fungus in the non-transgenic. So I think a pretty strong indication, this is 15% of the total number of genes in the native AM fungus response, is being replicated just by expressing IPD3. Now, the nature of this response is very different from what symbiotic plants do for their symbionts. But the fact that it's replicated is similar to how IPD3 works in symbiotic systems. Another interesting contrast to look at is this, which is the transgenic compared to the wild type when both are exposed to AM fungus. There's almost 1,000 differentially expressed genes. When we overlap that gene set with the native AM fungus response, again, quite a few overlap, 103. Of those 103, 89 genes go in the opposite direction in this contrast than in this contrast. And what that's telling us is that in the transgenic, expression of these genes is being pushed back in the opposite direction from what the native response is, right? So this contrast doesn't tell us what the response of the IPD3 transgenic is to AM fungus. This tells us how the IPD3 transgenic differs from the wild type. And it looks like one of the main ways it differs is in genes not changing their expression. in response to AM fungus. And we can confirm that in more detail. If you look at things that don't overlap, you see just gene ontology enrichment for really similar terms. So it's not like a bunch of additional conditionality there. So those things add up to informing what I think is the most interesting finding here, which is that when you expose the transgenic plant to mycorrhizal fungus, only 11 genes are differentially expressed. Happy to talk about exactly what those genes are later, but mainly in the wild type exposed to AM fungus, 500 genes are differentially expressed in response to the microbe. This actually makes a lot of sense in regards to the first two results we just saw. So we know that when you express IPD3 alone, about 15% of the native AM response is replicated just by the presence of the gene. So in that regard, the plants may not respond to AM fungus because they're already there. We also know from the second contrast we looked at that for about 17% of the 500 AM responsive genes in the wild type, those genes are prevented from responding the way they normally would. So that adds up to about 33% of the native response to AM fungus being accounted for in one way or another by expression of IPD3. Now, 33% may or may not be a huge number to you. You may be very concerned about the other 67%. I think the most likely reason that we see that mismatch is that those additional genes could be dependent on a core set. But of course, we're going to do that more rigorously and confirm exactly what's going on here. But this outcome is pretty remarkable. So we put IPD3 into these plants that have been missing it for a long time, and it really significantly changes what they do in response to a symbiont they haven't interacted with for a long time. So what we can do with that is a few things. We could really dig into the transcriptome, try to analyze it better, or get better information about each of the gene functions. A postdoc in our lab who really already did a lot of heavy lifting for the transcriptome, Melody Charles, has done additional analyses that we're using to help with this. We can also do more lab work in functional genetics to hunt down individual gene and protein interactions. We can, since this seems to do something pretty dramatic, try to put in more components of the common symbiosis pathway and see how they interact both with Arabidopsis and with IPD3 to probe how much further the potential conservation goes and where it may continue to break down. And for this broad implication of defense, with genes kind of flying around in different directions, with known functions in defense, to determine what that means for plant biology, probably the best thing we should do is just start exposing the plants to pathogens. We've actually done a bit of that. I'm just going to give you a hint at some of the follow-ups that we either have done or are doing. So first of all, this whole transcriptome that we did in Arabidopsis was actually mirrored in Lotus japonicus, which is a mycorrhizal model. In that case, the wild type has IPD3, and we added a mutant that lacks it. And another student in our lab, Megan Franklin, has analyzed that transcriptome, and we'll be looking for parallels, or more like mirror images, in the Lotus and Arabidopsis transcriptomes. We also have tried to look at defense. So we encountered a pathogen in our lab, Aspergillus niger, that just happened to infect wild lotus plants. And as other researchers have noted, we saw a protective effect of AM fungus. So this up here is the rate of necrosis in plants growing without mycorrhizal fungus. This down here is plants with mycorrhizal fungus. So plants seemed to die less from this particular pathogen when they were treated with AM fungus. But to really dig into this, we really need to work with a model pathogen, one that we know more about and that is probably a bit more virulent. We also tried to look at long-term interaction of the plant with the fungus by growing Arabidopsis and lotus plants in a special system where you have a divided pot, on one side of which is a plant supporting mycorrhizal fungi, providing them carbon. There's a membrane that lets the fungi cross and colonize the roots of plants that for one reason or another, because it's a non-mycorrhizal species or a mutant, cannot support AM symbiosis on its own. What that would let us do is see what the fungi really do in the root, look at more of a root phenotype and at different time points for gene expression. Unfortunately, when we try to do this, even the plants that were supposed to support the symbionts really don't get colonized. So what we're looking for is this, a bunch of intracellular symbiosis, arbuscules. The large majority of plants, every time we try to use that system, just have empty roots. So for some reason or another, we cannot establish the symbiosis in the exact conditions we had, something we really have to revise if we wanted to use it. So about 2 more minutes and then we're done. As Heike mentioned, I had an interdisciplinary project related to lupin, which is the only non-mycorrhizal legume. And I'm interested in it for a few reasons. One has to do with my interdisciplinary training in the Ag Biofuse program. Another is a collaboration my lab had with the National University of San Agustin in Peru, where lupin is a traditional crop. And it's not very widely used. I thought it would be good to analyze why. There's a few reasons. So lupin has great protein content. It also has pretty good oil content, but it actually isn't even one species. There's at least 4 domesticated lupin species. So the most common one that's highlighted in red is narrow-leaf lupin. There's 2 more relatives of it from the old world, white lupin and yellow lupin, and then the Peruvian or Andean species are called Andean lupin. So our view of what lupin agriculture even is a bit scattered. And we seem to keep domesticating it multiple times. So narrow-leaf lupin is maybe our most recently domesticated crop. This was domesticated starting in 1970 and to meet a very particular need in sandy soils of Australia. This is now the predominant version of the crop. And so I think our view of it has often been limited to the narrow context that this lupin was developed for. Their quality lagged soy a little bit, with the exception of Andean lupin. These 3 species have a little bit less protein and a lot less oil, so they may be less appealing for that reason. But they are very appealing for the fact that these grow in the winter. So in a warm climate like ours, you can grow these in the cool season. And not to put too fine a point on it, that's climate change stuff. The summers are getting hotter and more stressful. The winters are getting warmer. A crop that grows in the winter is of high interest. These also grow well on poor soils. So what I did is run a crop performance model for a sandy soil region of North Carolina using real world soil and climate data and simulated lupin yield. That's these yellow peaks. I also analyzed the growing costs if we were to imagine growing it, and found that lupin over those 15 years, if you look at this green highlighted square at the bottom, would yield you about an extra $600 an acre. So it looks very promising. But going into this is a really interesting fact about price. So lupin right now is priced bizarrely by being pegged to a fraction of whatever soybean sells for. And that has to do with its markets, not with its properties. So if we really want to change the fate of this crop, we have to think about not just the traits you could probably guess are important, like cold tolerance, yield, et cetera, but things that may change how it's viewed in the market. And that brings me back to this contrast, because the most ignored lupin, Andean lupin, is the only one where the product quality really matches soybean. So if, as a genetic engineer, we want to try to change what happens to a crop, in this case, I think we should focus on product quality, which would, for example, let us take Andean lupin, get it fit into additional agricultural systems outside of the area where it naturally grows, and take advantage of the fact that it can compete with soy on the market to change its competitive position, unlock its pricing, and maybe that way get it adopted in a way that you can't just through this incremental work. And of course, for lupin, there's a history of that. We keep re-domesticating it. And my view is if you could do this with narrow leaf lupin, with conventional breeding in the 70s, we can do it in Andean lupin now with biotechnology, faster. So that's what I'd like to do. I'll talk more about it with the committee. But for now, I'd like to thank all of you for coming. Thank my lab, current and past members, my committee, my funding agencies, and in particular, my family for being here. I'm really happy that my grandmother is watching today. She was the first generation in our family to go to college. And her daughter, my mother, the first generation to go to graduate school, is here too. So there could not be better footsteps to follow in. And with that, I'm happy to take any questions.
So if we start with questions from the chat, Bob Gottwals asked if you saw a PCA analysis on a slide, and if so, could you give that a little bit more time?
Yes, you did see a PCA. Thank you, Bob. And the reason I didn't talk about it is I had already started talking about the next thing by accident. So a PCA is a way of looking at really complex data with reduced dimensionality. And in this case, we feed the PCA analysis all of our differential expression information. And we ask, do the samples based on all of that information cluster according to the treatments and groups we applied? Meaning, do our treatments actually explain the overall variation in gene expression? And they do. So these circles are independent clusters. And across this bottom line, the first principal component, the axis that explains the most variation, they cluster by nutrient. So you'll see way over here, the low nutrient samples with and without the transgene are present. On the other side of that principal component, all of our samples grown on high nutrient are clustering together. On the second principal component, the plants cluster by gene expression. So down here at the bottom, you see members of line 308. Above it is line 310. And remember, line 310 had a less extreme phenotype than line 308. We also know from the transcriptome that this line expresses the transgene at about 20 to 25% the level of line 308. So it falls intermediate between the strong expressing line and the wild type, which is up here along this principal component. You may also notice there's not very clear clustering by fungus treatment on this particular PCA, even though we saw in the transcriptome results that there is quite an effect of fungus treatment. So that clustering may occur along a different principal component. If you imagine this coming out of the screen, that would be the third principal component. Or we may just be failing to visualize it here because of the effect of the first two. Hope that answered.
Okay, so I'll take the next one. So the next question is from a former member of this lab, Danielle Young, who graduated this year at MSU with a PhD. Hello, Danielle, I don't know if you can hear us. And what else? A very clever question. Did you or are you planning to investigate the nutrient uptake of IPD3 expressing Arabidopsis plants versus wild type?
So of course we did part of the transcriptome on low nutrient. We're a little bit hobbled by the fact that we lost the uninoculated plants. But definitely that is something we would like to look at. The response to phosphate stress is not one of the biggest effects in the transcriptome. So we might not hypothesize just based on that we'll see a big change. But going the other direction, we should expect that altering the phosphate status could interfere or change the effects of IPD3 expression because we know there's all of these co-regulated genes in models. So that's a very interesting thing to look at. We will look at it in the low nutrient transcriptome, and then hopefully follow it up in more detail.
Then there's another question from Aurora. Is the idea for the Andean lupin to make it an overwintering crop? And is that currently hampered by day length needs for flowering?
So in Andean lupin, it probably is. The idea in general, for me anyway, is to make it an overwintering crop. One reason that that's probably a universal idea is that they always take a pretty long time to produce fruit, all lupins. So you may as well grow them over the winter. Day length sensitivity is something that breeders and genetic engineers have a pretty good idea how to alter. So it's something, it's a good target to have. A bigger issue is probably cold tolerance, because even though winters are getting warmer, there's still going to be frost. And in a couple of field trials from the past 10 years, you still see frost damage as a major component limiting winter lupin.
Okay, last question.
So I'm wondering about the trade-off in lowering the host defense to have the symbiosis with the fungus, with the AM, but then being susceptible to pathogens. So isn't that a possible explanation for the loss of this?
So it's very possible. And in fact, speaking of one of the genes I saw, GPAT5, its near neighbor, GPAT6, has a homologue in mycorrhizal plants, which is one of those genes whose expression makes them more susceptible to infection by, in this case, the potato-like family of pathogens. So we have specific evidence that that could be the case. I think the difficulty is turning that into a general feature, right? That's really an issue, not a terrible issue, but for this transcriptome, saying we see differential expression of defense-related stuff is different from differential expression of defense. So we're going to have to do, I can't think of any better approach than actual experiments on defense to see how well these plants are defended from pathogens, to figure out, does the presence of IPD3 or interaction with AM fungus actually make them more or less susceptible to pathogens overall?
And with that, any really important urgent question?
I think this will be quick. Amara asked if IPD3 is also involved in nodulation signaling.
Yes, it is. If you put IPD3 min into a legume, you get a plant full of nodules with no rhizobia on there. Very important in nodulation.
And
Okay.
if we're out of time, I'll take the questions that are in the chat and forward them to Eli.
That would be great.
Okay,
Thank you very much.
so thank you, everybody. General topic, where cake and cookies and drinks. The committee gets a 5-minute break. Is that enough? You need 10? And then we meet back here for a little questions. Take a break, 10 minutes.
Thank you.
You're actually familiar with
I
lupins?
enjoy eating the looping during the looping session.
All right.
No.
I need to change something here. If you're on the video, stay on for a second.
Yeah, that's a good question.
So do people who are online see me?
Yes.
So I just wanted to thank all of you for coming. I know I wish we could have talked more or had you ask questions verbally, but I was to see you. Hey, Alex.
Great job, Eli.
Thank you. Yes, I also wish I had more time to go into the phosphate stuff, but yeah.
No problem. Thanks. Good luck.
For coming. Right, well, I'll say goodbye, everybody. Talk to you on the other side.